For most of human history, the earliest stages of human development were completely invisible. The first several days of cellular life occurred in total darkness, shielded from observation and measurement. Modern laboratory imaging has removed this physical barrier. We can now watch the entire sequence of conception unfold in real time, observing the precise chemical and mechanical steps that were once inaccessible. From this perspective, human development resolves into a sequence of measurable physics. We are looking at a programmed biological assembly driven by specific thresholds. Human origins are no longer a matter of speculation. They exist on a tangible timeline that we can track frame by frame. The timeline begins at day 0. The most controlled way to observe this starting point is through intracytoplasmic sperm injection, or ICSI. During this procedure, a single sperm is mechanically guided to penetrate the thick outer layer of the egg, the zona pellucida. This physical breach activates an internal clock within the egg. It initiates a programmed sequence of development that governs the cellular activity of the coming week. The microscopic entry of the sperm triggers a rapid biochemical chain reaction across the cell's membrane. Using advanced fluorescence microscopy, researchers at Northwestern University recently documented the visual evidence of that reaction. The instant a sperm fuses with the egg, a calcium surge causes it to expel millions of zinc ions into the surrounding space in a sudden, coordinated burst. This release of zinc establishes an immediate protective shield. It changes the chemistry of the egg's surface, helping to block additional sperm from entering the newly activated cell. Fertility doctors now use the physical brightness of this spark as a diagnostic metric. Because the intensity of the zinc discharge reflects the underlying health of the egg, it serves as a readout of the cell's potential to develop into an embryo. This flash is the inorganic signature of cell activation, predicting whether the developmental journey has the energy required to succeed. Following the spark, the genetic material from both parents combines in a process called syngamy. This is the moment the biological timeline officially begins. With time-lapse intubator technology, we can watch the subsequent phase of development, known as cleavage. This diagram maps the progression. Every 18 hours, cells divide on a strict cycle, 2, 4, 8. During this phase, a physical paradox occurs. Cell numbers increase rapidly, but overall size remains unchanged. They shrink with each division to fit the original protective shell. This confined fracturing continues until day 4, resulting in a tightly packed sphere of cells called a morula. This dense formation prepares the entity for its first major structural organization. On day 5, the process shifts from uniform division to structural organization. The cluster begins forming specialized cell groups, becoming a blastocyst. The blastocyst's interior architecture divides into 2 parts. Cells pump fluid, creating a central cavity, the blastocele, while the inner cell mass gathers at one pole to eventually form the fetus. The outer ring, the trophectoderm, will form the placenta and support tissues. IVF clinics rely on this specific architecture to evaluate embryonic health. They grade the density of the inner mass and cohesion of the trophectoderm to select the optimal embryo. By the end of day 6, the blastocyst has assembled a meticulously organized, gradable blueprint, fully prepared for the next phase of its journey. As the first week concludes, the expanding blastocyst must physically rupture and hatch out of its protective outer shell. Once free, the hatched blastocyst travels to the uterine lining. To survive, it must mechanically anchor itself to the uterine wall. Researchers are now using 3D bioengineering platforms to study this process. By utilizing synthetic gels that mimic the uterine environment, they can observe and measure the mechanics of implantation outside the human body. Successful implantation is the mechanical climax of the first week, anchoring the free-floating blueprint to a permanent life support system. These chronological events align with the consensus definitions of early human development. The first 14 days are categorized as the embryogenic phase. During this window, biological processes are primarily dedicated to building extra embryonic support tissues, creating the environment necessary for the embryo to develop later. Mapping this sequence has provided a new level of clinical precision in fertility. We can now measure and identify biological viability long before implantation occurs. The origin of life, once shielded from view, is now visible as a structured, measurable sequence of cellular physics.